Alrighty, thanks everyone for joining us today for the very first seminar of the semester. It is my pleasure to introduce Andrew Oates. Andrew is a professor at EPFL specialized in biological pattern formation. So to be more specific, his research focuses on the emergence of spatial temporal pattern at the tissue level from noisy cellular and molecular interactions. So today he will share with us some of the related work about how does a cell autonomous timer drive the wave pattern of the segmentation clock.
Okay. Thank you. All right, thanks very much. Thanks for the invitation. And I'm going to tell you about some work now that is been on the bioRxiv for a little while, but it's not published yet. So if you have any questions and feedback and stuff, would be much appreciated. So the project is, oh yeah, and please interrupt. I'm happy to take questions, particularly if there's something that's not clear or I can clarify as we go along. Please don't hesitate to interrupt me. So okay, here we go. So yeah, this work is a collaboration done between my group and with Frank Jülicher, group of Frank Jülicher. We started working together got a long time ago now, when my lab was in Dresden in Germany. It's now in Switzerland, but we've kept a good link together. And the work I'm going to talk about today was done by Laurel Rohde, she's an embryologist, by Ariane Bukowski, she's a bioengineer, and by Jose Negrete, and he's a theoretical physicist. So I'll try and point out the bits where they, where they had their main contributions as I go along. And really the work was driven by Laurel, following up on an experiment that she did, which I'll tell you about in just a minute. So the question that I want to tackle is to talk about issues of biological timing and how they give rise to patterns at a tissue scale. And first I wanted to just introduce these three concepts. One is sort of an ordering in time, where what's important in the system is that event A happens before B and so forth. But it doesn't really matter, or at least maybe we don't have any evidence that it matters, what the specific interval between A and B is. And then, so I would call that ordering. And then I would call a timer or timing, I would say that's when there's an interval between A and B that's important in some way in the system. So there's some rate from where A goes to B. And a good example there, sort of the analogy might be one of these sand clocks. But once the sands run through, then it stops. Okay, so that's irreversible. That's one way. And then we might talk about a clock. A good example there might be the circadian clock, a good example of a biological oscillator or clock. And that has internally repeating states. And of course, it's using energy to keep going, but it doesn't need to be reset from some outside mechanism. So I wanted to talk to you today about is the experiments we've had, which are looking at evidence for a timer and a clock in what we'd previously called sort of collectively as the segmentation clock. So let me take a step back and talk about the kind of biological question that motivates us. So this is a picture of a human skeleton taken from the back, and if you're a developmental biologist, as we are, you're interested by things like shape and size and number of body parts. And in this case, if you're interested in the segmentation, the segmental arrangement of the body, you're also interested in the timing of how those parts get made during embryogenesis. Now, we don't study that in a human, although there's some really remarkable work being done at the moment using cell culture models to study the human segmentation clock. We study that in a zebrafish. So here's a video of a zebrafish growing, and it's got a big ball of yolk that you can see in the middle, the light colored, and then this arc of cells around the outside is the embryonic axis. You can see an eye down here, and as the axis extends, it's from the tail bud down here, you can see that there are segmented blocks of tissues. These are called the somites, and they're forming rhythmically and sequentially as the animal extends its body axis. And, the cells that go into making each of these somites, these body segments, are initially contained in the tail bud. And so the question is, what's going on in the cells in the tail bud before they become the body segments? Now, the answer is actually that there's a segmentation clock in that tissue, in the pre-Semitic mesoderm, in the mesoderm in the posterior embryo here, which is green, that before those cells become somites, the pre-Semitic mesoderm. Now, this is a complex biological oscillating tissue. It was first discovered in the lab of Olivier Porquier by Isabelle Palmerin in 1997. And what I'm going to do today is I'm going to show you what we know about the zebrafish instance of this. It appears to be in all the vertebrates. And in fact, some kind of segmentation clock appears to be in most segmented animals, probably only not fruit flies. So, okay, two transgenes in the zebrafish, one of them in pink, and I'm going to use, both of them will feature today in the talk. One of them is in pink, and as each of the new segments is forming, you can see that the anterior part, the head end of each segment goes pink as it forms. And so this is the genetic marker of each of the new segments forming. And what's a little bit more interesting is this green signal, which these are both transcription factors, and this is a fluorescently tagged transcription factor that is giving rise, its expression is giving rise to waves in the tissue. And I hope you can see that there's a waves of green that are coming up from the posterior. They're moving through the tissue sequentially, and as each wave arrests in the anterior of the tissue, that marks the time and the position where each of the new body segments are formed. So there's a transition from a green oscillating tissue to a pink static differentiated segment. And each of those body segments will turn into muscle cells and bone cells and skin cells, and they will be the final segmented architecture of the body, like we saw in that previous slide of the human. Okay, so you can't see any cells in this time-lapse movie, but to see cells, we need to zoom in, and to do that right now, we're using light sheet microscopy because it's got low phototoxicity and lets us look at multiple embryos, and it's a really fantastic imaging tool. And this, I think, is really where the work of Ariane Barkowski has come in our lab, because she's been really instrumental in turning this fantastic imaging system into a useful way to ask biological questions by being able to deal with the data that comes out of the microscope. So, okay, so this microscope is made by a company called Viventus, who are here on the campus in EPFL. And what you can see now, so this, for orientation, here's the zebrafish embryo up in the top left, and we're focusing now on the cells that are green here, and that transgene is now white in this picture. And each of these little blobs is a nucleus in one of the cells in the tissue. So the posterior tip of the tail bud is out here. The pre-Semitic mesoderm extends along here. This is one of the waves stopped. I'll show them moving in a sec. And the newly formed segments are here, heading off towards the head up that end out of screen. So let me play this movie for you. And what I hope you can see is that you can see the waves of gene expression are spreading up, just like in that previous movie, from the tail all the way through the tissue, and they're arresting in the anterior of the tissue over here. And as each wave stops, the oscillation stops, the gene expression stops, and those cells then become a new segment. And when you look, you can see, I hope you can see that each of these nuclei is getting bright and dark and bright and dark, and sort of over and over again. And so the wave that you see at the tissue level it's underlain by individual cells switching on and off in a coordinated way with each other. So this segmentation clock is, a population of genetic oscillators, and the waves arise because those cells are, their oscillations are coordinated. They start and then they arrest as the cells differentiate. Yeah.
Question. So is it an impression, or the cells are moving also along the extension of the body axis?
Yeah, that's a good question. So it's a really important question, actually. So to first approximation, new cells are being added into the oscillating tissue from the posterior. And as cells stop oscillating and get incorporated into each formed segment, they are removed, if you like, from this block of oscillating tissue. So if you focus on the reference frame of the oscillating tissue, then cells are being added in one end and removed from the other end continuously. So yeah, in fact, there's an advective drift of the cells slowly through the tissue. If you like, the phase velocity of that wave is 8 or 10 times faster than the movement of the cells through the tissue, but both are going on. And that's actually really important because it means that each cell has a sort of developmental history where it starts out in the posterior of the tissue, and then over time, it drifts through the tissue until it exits the other end of the tissue with all the other cells that are around it. So I wouldn't say that the cells are migrating along the axis, but they're, they change their relative position in the tissue as it grows.
And the genetic or molecular environment, as they move along, or as the body axis, as it extends, this changes also. It's not the same being near the PSM, near the somite or at the beginning of the movement.
Yeah, okay. Super question. That's exactly what I want to tackle.
Okay.
Yeah, so for sure, the two environments are different, but how they're different and what effect that has, I think, is the key question actually today. Well, so we can finish there actually. Thanks, mate.
A quick question.
So, just to be a bit more explicit here, what you're seeing in the fluorescence channel here is this protein, the HER1-YFP fusion protein, that protein being translated and becoming bright, fluorescing. By the time it starts fluorescing, it's matured, it's already in the nucleus. Most of it is already in the nucleus. And then it's degraded. It's destroyed in the proteasome. So each nucleus gets bright as the level of transcription factor rises, and then it's destroyed, and so it gets dark again. Okay, any other questions about that?
Can I ask a question?
Sure, yeah.
I see like dotty signals in addition to nuclear signal. Is that like each allele, or do you know what they are?
Yeah, we're still trying to work out exactly what they are, but I think you're right. My hypothesis is that each one of those dots is the site of the transgene, because the transgene itself exists in multiple copies along the chromosome, tandem copies.
Oh.
And so because of the next diagram I'm going to show you, because this is a negative feedback oscillator, each of those dots marks the site where the protein is actually accumulating to do its repression. Now, I don't know that for sure, okay? But that's consistent with the data. There's usually one spot. Sometimes there's two spots. This is a heterozygous animal, and that corresponds to either before or after S phase, if that makes sense.
You use 10 copies of transgene?
I'm sorry, say again?
So you're using 10 copies of transgene?
Tandem copies.
Tandem, I see.
It means that there are multiple copies of the transgene. They've gone in the same orientation in a row at one location in the chromosome.
Got it.
I also had a question.
Yeah.
Within this tissue, should we view all of these cells as kind of being on the same footing, and this is a kind of intrinsic oscillation in the tissue, or is it being kind of, is there some driving on one side that's kind of almost like an extrinsic source, and then it gets passed along through the tissue?
Yeah. So, should we think about it like an acoustic wave where you need a driver, or is it more a kinematic wave? So we think it's a kinematic wave, and the first evidence from that actually came from the experiments that were done in the Pourquier lab by Palmerin, because she used the chick embryo and did a transection of the tissue and showed that the downstream part of this tissue, at least for one cycle, appeared to change its pattern. Now, they didn't have any transgenes back then, so these were carefully inferred from time samples that were operated on and fixed and then the gene expression was examined in it. But she was exactly right. And what I'll show you next is some evidence that the individual cells oscillate quite fine when we take them out of the tissue. Okay, so any more questions here? It's good, actually. You've hit most of the key questions for the. Okay, what's going on in individual cells? So this transcription factor is an example of a family called the basic helix with helix transcription factor. It's not really so important at all for this talk what that is. Those proteins form dimers and homo heterodimers, and they have these flexible helices that engage the DNA in a sequence-specific manner. So they're typical transcription factors. They nearly all of these in this class, the Hess-Her class, they are transcriptional repressors, and that is important because that's our current model of what gives rise to the oscillations. You can imagine having one of the genes here, the Hess or Her gene, it's transcribed, the mRNA is translated into protein, dimers form outside the nucleus, they re-enter the nucleus, and then they can repress their own transcription. And so if the half-life of the mRNA and the protein is relatively short, importantly, it's balanced with the delay it takes to go around this cycle, then this circuit could give rise to stable oscillations. So there's lots of evidence to support this model. I still think we're missing a really good watertight by physical demonstration that by tuning the parameters in the system, we can step across from a non-oscillating regime into an oscillatory regime, vary the frequency in that regime, and then exit the other side of that volume in parameter space and stop oscillations again. That hasn't been done yet. There's actually a student in the lab who's trying to do that. I wouldn't be surprised if that happens, but I think we still don't actually have the proof by synthesis that that's what's going on in this system. Okay, that's a side note, but okay, so what have I told you so far? So segmentation clock is a tissue level rhythmic pattern generator. It's got cellular oscillators in it, which there's some sort of negative feedback loop running in those cellular oscillators. The oscillations are coordinated together locally. So there's some local synchronization between cells, and that we think is driven by the delta notch signaling pathway. I'm not going to talk about that today, but if there are questions, I'm happy to say what we've learned or what we think is going on. But oops, I'm pressing the wrong direction. And then of course there's these tissue level wave patterns that they are now occurring at the length scale of hundreds of micrometers across the whole tissue. And what we want to do today is somehow connect the behavior at this level of the cellular oscillators with the behavior at the tissue level of the wave patterns. Now, this is kind of important because this is important for the animal, because when the segmentation clock doesn't work, for example, when the cells are not synchronized, the output into the skeleton is that there's a strongly disorganized skeleton. And so here are some examples of fish, mouse, and humans that can't synchronize their oscillating cells. And in each case, the skeleton is sort of mispatterned and deformed. And this is an example of congenital scoliosis. Okay. Now, let's go back to this question about the pattern in the pre-Semitic mesoderm. So what generates the transitions here? So we've got oscillations in the posterior. We've got these oscillations of individual cells. We've got waves. And then we've got a transition to differentiated state in the anterior. So what's driving that? Now, the first question is, what is you saw the cells are oscillating, but what's happening at the individual cell level? And so we can go to those movies that I showed you before, and we can follow individual cells from the tail bud. Now, this has taken us many years to be able to do this, but we can pick up individual cells in the tail bud, and we can track them over many hours as they drift. You look at this red dot as it drifts through the reference frame of the tissue and finally becomes incorporated into a finished somite. And as you can see there, is just a small number of cells that are going into the same somite. The oscillations, they're oscillating in the tail bud with low amplitude, and as the cells exit into the pre-Semitic mesoderm, the amplitude grows and they actually slow their oscillations at the same time. It's important that they slow their oscillations because the slowing oscillation creates a phase difference along the tissue. Now, the cells then up here, when they arrest right at the anterior end of the presemitic mesoderm. And then in that same part of the presemitic mesoderm where they're arresting, they're doing their last oscillation, that's when they're switching on this marker of differentiation, MESP. So that's the other transgene. And so if we follow individual cells, which have both those transgenes, we can see the last peak as the oscillator turns off up here. And then in those cells, here are the corresponding traces for the marker of the anterior cells in each of the segments. So there's the question here is, how do the cells know when and where in that tissue they should have their last oscillation? Yeah.
Oh, just a question really related to what you're saying, but related to the previous slide.
Yeah.
I was wondering if you align, so the different green lines are different cells that you marked.
Yes.
And you align them with time. Are they also aligned with space? That's what I was wondering. Did you try to align them with position along the presemitic mesoderm?
Yeah, all of these cells go into the same segment.
They go in the same segment. Okay.
Yeah. So in other words, I could have said, okay, what we did is we went to the 28th somite and we backtracked those cells until they were in the somite. So we know that these are actually neighboring cells in this somite. They're very closely, they're within, I guess, 20 or 30 microns of each other. And so that's their time trace over the whole, the time course from when they're in the Touboul.
So it's fair to assume that they're at the same location during their migration through the PSM.
Yes, and in fact, that's true, that while cells are in the tail bud, while cells are in the tail bud, they actually mix quite strongly. The tail bud has, I guess, fluid-like properties materially, which is one of the reasons why it can change its shape and move so dramatically. But as cells, so I would say, although I don't put my hand in the fire for these particular traces, but back here, I wouldn't expect that these cells were necessarily spatially very close. And they could be changing their relative positions with each other. But as cells come out into the presemitic mesoderm, sort of level with the posterior end of the notochord, they stop moving relative to each other, or at least the movements slow down dramatically. And now they basically hold in place with respect to each other all the way along that tissue. Yeah, so we've sort of got a mixing zone, and then we've got like the concrete mixer has finished and it's poured the cells down, and now they're more or less stuck in place. Of course, when they make the somite, they have to undergo lots of interesting mechanical changes and gripping their neighbor and changing their shape and stuff, but they don't rearrange their position dramatically like they did back here.
Thank you.
Is that okay?
Yeah.
So here's the question. So where's the information in that system that the cells are using to slow these oscillations and to stop the oscillations, and for this differentiation marker to come on, this MESP, which will actually be upstream of some of the muscle identities, for example, in the cell. So the ideas in the field, there's a couple of different interesting ideas in the field. Actually, there's several ideas in the field about what might cause the slowing of the oscillations and what might cause the arrest in a particular position. One of the ideas, and it's I think the first one in the field, it goes back to this classic idea in developmental biology of positional information guided by gradients in a developing tissue. We know from many labs that FGF and Wnt, which are secreted signaling molecules, proteins actually, are being produced in the posterior of the tissue in the tail bud, and we know that they can form a signaling gradient across that tissue by a number of different readouts, molecular readouts. We also know that retinoic acid is produced by the formed somite after it forms, And it can also spread in the tissue, and there are estimates being made about how far back into the presomatic mesoderm it spreads. And so one idea is that, you know, the cells might use these gradient systems to go from a sustained oscillation to slow down their oscillations into a rest. And they may also use that to know where in the tissue that, you know, that arrest happens and where they should now become differentiated, become determined as muscle cells, for example. For the aficionados, you know, so this is some kind of a wavefront in the tissue. So that would be the wavefront of the Clocken wavefront. But, and this comes back to the question you asked before, there's another thing going on in this tissue. In the reference frame of the tissue, there's also a cell flow. Okay, so cells are coming out of the tailbone and they're drifting through this tissue, moving more anteriorly. As that, you know, as the tissue grows posteriorly, cells are always being added from the posterior and they're drifting through that tissue. So when we think about this coordinate system, if you like, of the gradients, the cells are continuously flowing through that system of gradients. So this is, you know, this is sort of unlike a bicoid gradient in the early Drosophila embryo, where although the cells are dividing, they're not changing their location in the gradient system. They basically stay where they are. So this is quite different. It's got a moving cell field. And the cell field moves with respect to the. So it could be, right? So it could be that the cells actually pick up something in the posterior, some state, some molecule, and they carry that with them as they go anteriorly with the cell flow. And so they might be using some sort of intrinsic information that they have, right? So, you know, is it intrinsic? Are the cells acting like radio receivers and picking up different signals that they can get? Or is it intrinsic? Could this be some sort of gradient by inheritance? And considering and it could be a mix of the two. It could be some influence of the two. Now, it's been difficult to try and tease this apart because these candidate signaling molecules, FGF and Wnt and retinoic acid, they do lots of different things in early embryos. They control cell division, cell migration, as well as patterning. And so it's been, I would say, it's been hard to do a definitive experiment to try and tease these apart, although there's lots of evidence, for example, that if you change Wnt or FGF or retinoic acid signaling, you can certainly change the length of a forming segment briefly. And that would be consistent with them having some ability to change the position where the cells are becoming determined with respect to somite. So there's no question they can do something in the tissue. The question is, what are they doing in the tissue? So, you know, if you want to ask this sort of question, what you'd like to do is you'd like to be able to get the cells out away from these tissue level gradients, and then maybe reconstruct the gradients or give them different signals and stuff like that. So this is a task that Laurel took on some years ago to try and work out a culture system where we could recapitulate the native sort of conditions without any growth factors, without any serum, without any inhibitors. What's the minimum state? And then we could add things back to it. So here's the experiment, very simple. It's possible to dissociate the cells. In this case, I'm going to start with dissociating cells from the part of the presemitic mesoderm just after the cells have entered from the tailbone. So they've just come out of the tailbone, starting down this pathway of slowing their oscillations and differentiating. We mechanically dissociate them, and then we plate them at very low density on a dish, and then we do time-lapse microscopy on them in L15 medium, which is basically a very simple defined nutrient medium. And we plate these at very low density, but everything I'm going to tell you today is also takes place after serial dilution. So it's autonomous behavior. So here's the question is, what can the cells do? Can the cells differentiate under these circumstances, or do they don't oscillate at all, or maybe they keep oscillating continuously, like a persistent oscillator, for example, like the circadian clock? So here's what happened. Here's just an example of a cell. So we had a bet in the lab about what would happen. And here's an individual cell oscillating green, and then it stops oscillating, but it didn't die, it differentiated. And so this cell here, you can see plotted out on the right here. And, you know, that looks quite a lot like the behavior of the cells I just showed you in the embryo. And this was a striking experimental result. I actually lost a bottle of wine because that's not the outcome that I bet upon. But so cells oscillate, then they stop and differentiate in culture. And they do so in a very, very, in a reduced, a simple culture, basically almost as we could imagine. Now, so that's a pretty striking result. The cultured cells, they recapitulate the embryo dynamics. So they slow the cells. So if we compare, they both slow and increase their amplitude. So we can compare the successive period and the successive amplitude, and we see that the majority of the cycles increase amplitude and increase the period. They have quite a large variation in the number of cycles that they'll do until they stop in culture. So here's an example here. This one's doing 4, 5, 6. This one's also doing 6 peaks before it stops. And when we compare that to cells that we follow from the equivalent region in the embryo that we cut out for that culture experiment, we can see that they also slow and gain amplitude. But what's interesting is we see that, this is perhaps not surprising, we see that they're much more precise in terms of the number of cycles they do before they exit. And that's not surprising, right? Because I just showed you that all of those cells are resting at almost exactly the same spot in the embryo. So that's a really interesting observation. 2 observations. One is that qualitatively, those cells are doing really similar behavior without any extrinsic factors. And the wave patterns that follow from the slowing, the coordinated slowing of the oscillation, that means that the wave patterns are actually generated by this slowing. So, active signaling from neighboring cells or spatial gradients is not required to get the slowing and differentiation. So that although there is a signal gradient in that tissue, that can't be what's driving the behavior that we're seeing. It does give good evidence for a gradient by inheritance in this system, where the cell flux in the tissue is carrying some information from the posterior. And suggests that there's some kind of internal timer or timing mechanism in those cells. So this positional information in the presemitic mesoderm, we think, at least in the zebrafish, maybe it's better to think about this instead of a positional axis as being a temporal axis. And that's the relevant sort of thing that we should be concentrating on. This wavefront, actually, all of its behaviors exist in a single cell. So, right, so now, can we see the signature of a timer? And there's 2 kinds of experiments I wanted to talk about here now. So we've talked about what a clock could be like. And now we want to say, okay, is there some sort of timing mechanism present in those cells? And what sort of evidence could we get about what that timer could be? So I have to say now, I don't know the molecular identity of the timer. We have some candidates. So I want to talk about the kind of behavior, the kind of properties that timer would have. And that will let us think both about what we ought to be looking for its identity, but also what the role of extrinsic signals could be acting onto the clock. Okay, so that's where I want to go next. Now, one prediction you might have of a timer that might be started or be running as cells exit the tail bud is that the more anterior along the tissue where we would take cells, the shorter time left the timer would have, and so the quicker the cells would exit their program and differentiate. And okay, I just realized I've got the word tick here, but I don't mean tick, because I'm not trying to convince you that the timer itself is ticking. I'm going to use the metaphor of an hourglass running out. Okay, so in fact, so Laurel did this experiment where she cut out very small groups of cells from different spatial locations. She compared their behaviors together and then measured how long it took for those cells to exit the program, to have their last oscillatory peak and to differentiate. And so what we saw was behavior that was consistent with a timer. So cells that were taken from the posterior, they take much longer to exit, to do their last peak. And as we took cells further and further anterior along the PSM, they exited quicker and quicker the program. So this is consistent that the cells in the embryo have behavior that's consistent with an intrinsic timer in them. Okay, so what else could we learn about this timer? And then we thought, well, what about looking at the behavior of the clock itself. Because this sort of noisy but stereotypical increase in amplitude might be a signature of the timer, right? Because we see and what we learn from that? So we decided to use a simple theory of a negative feedback timer to see what properties we could infer about the timer from the waveform itself. So we think that the clock mechanism is quite complicated. So we wanted to try and choose a really simple representation of that. Now we're just going to have a gene that produces a protein, and then the protein is degraded sometime later. And for that, we use a delay differential equation. And we're certainly not the first people to use delay differential equations for this kind of problem. I think Underheiden and McKay were the first people to use this. And so here we keep track of the amount of protein that we've got of this HER1 repressor protein. The protein, of course, has a half-life. It gets degraded with some time constant. It can repress itself, and we use this we use this term here to denote that repression, and it does so some time interval after it was produced, and that mimics the time to go around the cycle. And then there's some production term, which would give you the production rate of the protein in the absence of repression. Okay. Now, in the past, we and others have actually done quite a lot of work thinking about the segmentation clock using equations like this. And we've played with the degradation rate and the time delays. We've thought about different kinds of noise in the system. What we haven't done is we haven't really played with the production rate. We've tended to keep that constant. And what I want to do now is talk about what happens when you play with the production rate, because that's actually what we think is driving this typical transient dynamics in the clock behavior. Okay, so what's the timer process that could be driving the clock feedback loop? I'm going to propose that it's a change in the production rate in the clock, and so that's what the timer is affecting. And so we introduce into the model a timekeeping factor or some sort of timekeeping process. Because we don't really know anything, we don't have a molecular identity for it, we don't have something in our microscope images that we can measure, we're going to try and keep that simple, phenomenological, as simple as we can. And we're just going to insist that it does three things, really. It's going to accumulate over time. It's going to reach some threshold, then it's going to decay away with some time constant. To get the kind of variability that we see in the data, we overlay on this sort of linear, very simple process of the timekeeping factor. We add Brownian noise to that to get the amount of the timekeeping factor that's present, and that works very well fitting to the data. So that's I don't have a sort of microscopic justification for that, except that it fits the data very nicely. So that's what we use at the moment. So what's the difference between having a persistent oscillator with noise and a transient oscillator, an oscillator showing transient dynamics? So I'm going to illustrate that now with two different biological clocks, the circadian clock. I guess you've all had jet lag, so you're all intimately familiar with the circadian clock being out of sync with your local daylight cycle. But here are fibroblasts grown in culture, and they oscillate continuously over this 40 days, this data. It's not ours, it's from Liza et al. They very generously shared us traces of their data, which was published. And using our model, we can take a constant production coming in to the clock system, and then with multiplicative noise in the clock circuit, so different to the Brownian noise I told you about just before, multiplicative noise in the clock circuit, we actually get a very good fit to the statistics of the oscillations. As a persistent oscillator. What we see in our experiments of the isolated segmentation clock cells is a very characteristic transient dynamics with a rise of amplitude and an increase in period, and then a sudden fall. And so for that, what we do is we take the production to to be what we'd call a noisy ramp. Okay, this is what I just showed you in the previous slide. And then when we look at what the clock circuit will do when it's driven by this noisy ramp, we again get good agreement to the data that we see, and we can then fit that using the statistics of the oscillations to try and understand more about what the ramp's properties have. Now, so we can, using the distribution of the amplitudes and the timing in the waveform, we can then infer what the clock should look like, at least what this change in the production should look like, which would be the output of the timer. Okay, so do we have any candidates for what could generate this kind of noisy ramp? Our best guess at the moment, and it's a guess, but I'll share it with you now because it gives you some insight into how we think about the problem, is a transcription factor, another transcription factor, which is expressed in the tissue and forms a pattern in the tissue. So there's a class of genes called the Tbox genes. These are transcription factors. When we look at the pattern of their expression in the presemitic mesoderm, the tail bud in green and the presemitic mesoderm, there are three that I'm looking at here. We've made antibodies to each of those three in this case, and we're now making transgenic lines to be able to follow their expression in real time. We can see they have this really striking distribution. So in green, we have a no-tail. It's only expressed, the protein only accumulates in the tail bud. So this is where the cells are bundling over each other and moving posteriorly. The cells that exit the tail bud, they start to express Tbox 16, which you can see here in this part of the panel. And there, that forms a smoother gradient across the tissue. And there's one gene, one protein, Tbox 6, which is expressed in cells after they exit the tail bud. It makes a ramp and then it switches off just as the cells are differentiating. So we want to focus on this. We became interested in this gene because of this distribution and because there are binding sites for Tbox 6 in the HER1 promoter. So we've made a transgenic line where this protein is expressed in the embryo. And I think we played this movie. Yeah, you can see that, remember there's a cell flow here. So cells coming out of the posterior, they're not expressing this gene not showing this protein in the tail bud, but as they flow anteriorly, they express, and as they exit the tissue, finally in the anterior, they switch the gene off. And Laurel's been able to measure from some of these cells, and this is preliminary data, that in culture, we do the same experiment now I showed you before, but we do them from animals from this transgenic line. You can see that the accumulation of this signal goes up and then it switches off again. So it has some similarities to these noisy ramps that I was showing you that we infer from the model about what the timing activity should look like. So, okay, so is this the timer? I don't know. But we think this would be a good candidate to start thinking about the identity of the kinds of molecules in that tissue that could be providing cell-intrinsic timing activity onto the clock. So, this is interesting, again, coming back to the, what does the animal care about this? Well, we know that when Tbox 6 is mutated in mice and humans, it actually causes congenital scoliosis. And here's an example from mice. This is work done by Diana Chapman, and then a series of papers now, much more recently, looking at human mutations that cause this congenital scoliosis. So we think this is an interesting candidate to follow up with. But again, I don't have any proof that this is actually the timer, of course. For that, we need to change the property of the molecule in the tissue and show, and the cells, and show that these transient dynamics either finish too early or finish too late. Okay, so we think that single cells have a clock driven by a noisy timer. I've showed you some of the evidence that we've got for that now. And that this gives rise to the characteristic transient dynamics that we see both in tissue and in culture. The role of intrinsic signals. So, there are the extrinsic signals, the role of extrinsic signals. There are extrinsic signals in the tissue. I think the surprise for me in this coming from this work is that they don't drive this program of the transient oscillations and the exit. That appears to be cell autonomous. It appears to be an intrinsic program, but they are doing something, and at least the sum of those signals is making the precision of that intrinsic program higher. So there's a tighter distribution of the number of cycles that cells do before they exit, for example, and it's quicker. So extrinsic signals, the sum of them, maybe the integral of them that the cells experience in tissue, is to hasten and sharpen the exit of cells in the tissue from that program. So we could think about then, if we have this concept of the timer, we have some candidates then that the extrinsic signals, we would argue, are not acting directly on the clock molecule, on HER1 or one of the other transcriptional feedback oscillators, potentially in the system. What we think they're doing is that they ought to be now acting on the timer, and then this oscillating system is just a slave downstream of the timer. So now we want to see, can we find evidence where, when we use these kind of signals for change in the accumulation threshold or the decay, by looking at the difference between cultured cells and cells in the embryo, we can see what happens to the accumulation threshold and decay. We can compare those two concretely, and we know that the accumulation and the threshold are much lower, and the noise is much, much less in the tissue, but the decay rate doesn't change. So that's one way of thinking about how the signals are doing. Yeah, Kana, I can see you.
Yeah, I have a question. So I think between single cell culture and embryo, you can think about collective cell that's cultured in the dish, right?
Yep.
So this feature could come from just the result of collective behaviors of cells, not necessarily from extrinsic signal. So have you tried?
Well, okay, so but maybe that's a definition, question of definition. So for me, an extrinsic signal is something that the cell doesn't itself have. So of course it could be supplied by a neighboring cell. It could be supplied from a cell that's a long distance away. This doesn't distinguish that. I'm agnostic about that.
I see. Okay.
Yeah.
Thank you.
So I used the example of FGF and Wnt and retinoic acid, which could be acting at a long distance, right, as a gradient, but it could be something like delta notch signaling. It's not, but that's a good example of another signaling system that we know is present between the cells. It could be the fact that cells are altering each other's mechanical environment, and that would be consistent with, for example, with some some results that Alexei Ubo had, for example. But I think what's very clear here is that whatever those are, collective, that can be acting in the tissue or in one of these culture models, at least for the zebrafish, they're just not necessary to produce the basic behavior. That's entirely cell autonomous. I'm not saying they're not acting in the tissue, but the cells doesn't care. It just doesn't care. Look. It doesn't mind. It does this behavior of slowing and differentiating exactly the same. Sorry, not exactly the same. Qualitatively the same, but quantitatively differently. So how long does it go? That's different, right? And then the collective activity of the signals that I guess are present in the embryo or in these cultures, that then is, I would guess, this is a hypothesis, right? But is now causing all of the cells with these noisy timers to do the same thing at the same time. And that's important. Otherwise, the animal gets scoliosis and it's in big trouble, right? So but the question is, what are those collective things doing? What are the signals doing? They're not producing this behavior, but they're probably modulating it to get it to be precise enough to do the job for the embryo now at the tissue scale. Now I don't know if that balance is going to be the same in mice and human cells. I think that's a really interesting question. I don't know. We haven't done any experiments on mouse and human cells or any of the other, let's say, warmer, furrier vertebrates. So this is just what we know about from the zebrafish system.
I see.
Does that make sense?
I think my question is, have you can you dissociate cells and then reassociate them and culture together
yeah,
and see
yeah,
the
okay.
difference?
Yeah, that's okay. That's a super question. So we make these sashimis where we can get cells together afterwards, and so dissociate them separately, bring them back together. A lot like the first protocols that you know, the MPSM, monolayer PSM that I think
I see.
Risos and Alex from Alex's lab published, and that works also in the zebrafish actually. So
that's so cool.
Yeah, so we see these initially. There's some sort of broad level oscillations, and then there's an onset of wave-like behavior, but we're only just characterizing them at the moment. So yeah, I think that does happen. So maybe I think that's going to be a great way. And so, for example, we can, even with two cells, we can see two cells synchronize with each other in the dish, synchronize their phase, but it's not enough to get them to synchronize their exit. Right. So signaling. That's why I'm saying I don't think delta notch is what's causing the increase in precision of exit of the program. Because we can see cells that will synchronize their phase. That's what delta notch is doing, but it doesn't mean that they'll exit the program at exactly the same time. So, right? Yeah. So there's something else, right? There's something else going on. I think that's really going to be very interesting to try and find. So, of course, I'm telling you some. This is sort of high level stuff. Obviously, Kana is a very high level operator here. So this is. Let me make a sort of broad conclusion, if I might, then, and then maybe we can. We think at the moment, at least in the zebrafish, that extrinsic signals, meaning signals from other cells, or either from contact or from long distance signals, or other signals might be present in the tissue. I think they do have a role controlling the clock, but I think that they act indirectly on the clock through the timer, and so by modulating this timer is how these collective behaviors and also instructive signals acting in the embryo, long-range extrinsic signals might be acting to control the precision and specific output of the behavior of these transient dynamics. Good. So, all right, we think in the zebrafish that there's cell-intrinsic timing is actually driving the wave pattern of the segmentation clock. It is controlling the slowing and stopping of the oscillators and also the differentiation. So we think this is very good evidence that the cell flow is really important for some sort of intrinsic property there. If there's an intrinsic program, how does it vary with position in PSM? I showed you that data. How does it change with developmental stage of the embryo? And when is that intrinsic timing locked in, or can it be kicked off again? Can it be restarted? We're doing experiments to try and find that out. How is it modulated by the intrinsic signals we know are in the tissue, chemical signals like these molecules I talked about to you before, substrate mechanics, cell-cell signals? Could these be ways that modulate either the duration or the precision of the program? And then, of course, what is the identity of a timing mechanism in the cells? Is there one timer? Are there multiple timers? We don't think it's a single component decay alone. We don't think it's the clock, because when we look at embryos that don't have a functional clock, the cells exit differentiation on time. We favor at the moment some sort of transcription factor cascade, maybe like neuroblast timing from Drosophila, if that means something to you. We think Tbox 6 could be a component of that, but we don't have any definitive evidence yet. So that's it. I'm going to stop now. I've been talking for 52 minutes and 50 seconds. If you have any questions, please, I'd love to hear them. Yeah, feedback's really, really welcome and necessary.
Well, thank you so much for such an interesting talk. So please feel free to ask Andrew questions if you have any.
I have a strange question. I mean, that's really fascinating. I mean, that was very good and really thought-provoking. I was wondering with what you said, would it be something we can imagine, but it is just, I mean, in terms of dynamical system for one cell, we just see the transient after an initial condition in the tail bud, and then it's just a relaxation to another equilibrium. And to imagine that there is no signal, in fact, no interaction, nothing. Cells are synchronized just because they started from the same initial condition and just relax with the same. I think, I mean, it's just what occurred to me when you presented. I was wondering, you know, would it even be possible? How could we do
it you know, I think that's possible. I think that the key is to ask what kind of dynamical system generates that behavior as it relaxes in the circumstances that you're talking about to a steady state. And we haven't really got any good candidates. I wouldn't say we've exhaustively surveyed the scene. And if we take this model that we use, then it has the sort of properties of being you know, driving across some bifurcation, being sustained for some time, and then dropping away again. So at the moment, that's my favorite model. Does that rule out what you're saying? No, it doesn't, right? So yeah, if you know, we looked at a couple of the other conditions, but they, you know, it's in the details of what the period and the amplitude do, right? Because as different systems go towards the steady state, they generate typical behaviors, and we don't see a good match from the ones we've looked at. But you know, you know, maybe we just haven't thought about the right kinds of systems.
I have another. I mean, if nobody has another question, I have another one. So I read the paper by Hubo and Pourquier, and they suggest an alternative model to the clock, to an oscillator, which is assuming that the gene expression is an excitable system, or in other words, like it doesn't have an intrinsic periodic orbit in terms of dynamical systems again, but it has a fixed point, but very different returns to equilibrium. So I'm wondering if that, would that make a difference in what you, in what you're looking at? And I mean, how would that, well, do you have any opinion on that? I mean, I mean,
that's, so, you know, I guess to first approximation, that's simply inconsistent with the data that we have. Because typically I'm thinking about now, you know, dictystelium, cyclic AMP oscillations, or sort of excitable neuronal systems, or other plenty of examples, you know, that's a collective effect. Now, it could be, you know, it could be that these cells are they're having some sort of threshold, sort of readiness to fire, that's being manipulated in some very, you know, interesting and unusual way by the culture conditions. But it's sort of a bit of a boutique explanation for the data, right? So I think that I think it's probably not the case in the zebrafish that's the way, that's the dynamical explanation. Right? I think it's probably different. Now it's interesting whether that's the right explanation in the mouse. That's another question, because I don't think that the data that has been published, at least, rules out a number of other models, certainly. So it could still be the case that it's not that case. It's not true. But it could be that there are a number of genetic differences in the sort of networks that are controlling the system in comparing mouse and chick, for example, but particularly strongly different comparing zebrafish and mouse. I mean, the usual suspects, they're delta notch signaling, these Hess-Her genes, FGF, but they're wired slightly differently. For example, Wnt signaling oscillates in the mouse and it doesn't oscillate in the zebrafish, and same is true for FGF signaling. So you could imagine, I could very well imagine that across evolutionary time, different dynamical systems have actually been explored and then selected and locked in for different properties that they give to the embryo that at the moment for us are not clear at all why one would be an advantage or not, what selective pressures acted, right? So yeah, I actually think that's a fascinating question. It's one of those really cool areas in, I think, sort of developmental biology, evolutionary developmental biology, where at one level the system looks identical. You have these sort of blocks of somites forming. There's some sort of clock there, and then you dive in a little bit deeper and you go, actually, not all the genes are the same, are they? And maybe even the dynamical systems, which is really what's being selected, right? They're not exactly the same either. So I think it's fascinating. Right?
Very interesting.
I have an impression that even if you use delay equation, if you play around the parameters, maybe you can make excitable system.
Yeah,
so it might be because of the circumstance of the cell.
Yeah, I don't know. It may be that you could, well, yeah.
Yeah, because the same equation can produce both dynamics.
Yeah,
depending on the parameters. So
that's right. So yeah, yes, you might not even need to think of some completely different way of writing it down. You might just different parameter spaces. It's slightly
I would
different.
assume your system is a good system to test that, because you can tweak the parameters.
Right. Certainly try and look for collective behaviors, although it's very difficult experimentally. When a cell stops oscillating, you have to be careful that you can then bring it back again. Otherwise, there's lots of reasons why a cell stop oscillating. You want to make sure it's the right one. One thing I did want to say, I asserted before that, at least in this model, by varying the production in a time-dependent way, we got very good fit to the statistics coming from the clock. I just wanted to point out that when we try time dependence of the delay or the half-life or the binding constants, the binding constants in that Hill term, none of them even qualitatively recapitulate the observed behavior of the cells. So I'm not saying that some complicated mix of all of the different parameters in the equation might be able to give rise to the observed behavior. But if you just change the production, you get really good fit, and none of the other parameters perturbed in isolation even give the qualitative behavior that we see with the cells. Actually, it's sort of a bit of a dynamical systems homework to go and see why that would be true.
Like in what sense is the behavior different?
If you say, okay, if you would hold the other parameters constant and say, okay, now I'm going to use the delay as a forcing parameter, and I'm going to see whether if in a time-dependent way I cause the delay to change, can I get the behavior? Can I get the clock behavior that we see? And of course you can get the oscillator to show transient behavior by varying the delay, but it won't have the statistics it won't have the relationship between the amplitude and the period and the up and down cycles that the data has. So that's what I mean.
I see.
Yeah.
Also, I have this this question about scaling.
Yeah. Okay.
Yeah. So I think as development proceeds, PSM size becomes much smaller, right, in zebrafish?
Yes, absolutely.
So the number of oscillation one cell experiences becomes less, right?
That's right. That's right. So
yeah. So do you
So
see
this is
that
what
behavior in your system?
developmental changes, right?
Like, have you
So,
tried dissociating cells
yeah, of
from
course we
different?
have. Yeah. Again, I don't have definitive evidence there. So I would say this is what I'm going to say next is certainly preliminary. So maybe I'll make this. Let's see. I propose like a thought experiment to see if it's what you're talking about. At early stages in development, the pre-Semitic mesoderm is really long, right? You saw that in those first movies I showed you, and it's true what you say. Kind of cells then take a long time to go through, and so they do lots of oscillations by the time they exit. Whereas by the end of development, the pre-Semitic mesoderm is much shorter. It scales down from something like it shrinks down from something like 700 microns to about 150 by the end of zebrafish segmentation, and the number of cycles also drops dramatically and correspondingly. It's not strictly linear because the phase pattern changes through developmental time. The phase pattern itself does not scale. So, so there's not like a one-to-one relationship there, but still it gets much less now. So you would say, okay, if the number of oscillations that it's going to do, that could be set by, let's say, changing Hox gene expression in the tail bud by some condition in the tail bud. And so if I pull cells out of the tail bud at early stages, they'll go for a long time in culture. And if I pull cells out of the tail bud at the end of development, they're only going to do a few cycles. So we haven't systematically surveyed all right across development, but we did pick 2 stages that have, you know, a very different number of oscillations in the tissue, and they have the same number of oscillations in culture.
Oh, even from different developmental stages.
So a 6 somite stage and a 20 somite stage. And they, at least the preliminary evidence is we can't distinguish which stage we got them from. Okay.
Oh, I see.
Right. So there may be differences, but at least you count, you know, putting that distribution of the number of cycles. So that's preliminary evidence. I think the direction it points is to say that maybe that's part of the role of these extrinsic signals is actually to control, you know, as I was saying before, to control the number of times an oscillator would cycle before it exits. That would be a good role for an extrinsic signal. So maybe that's, you know, so where exactly is that? Maybe that's being set by these signals, but you know, that's a hypothesis.
Yeah. I just thought if the scaling property is intrinsic feature of the cell, it would be really cool, but
yeah, that would be
it's very new.
That would be mind-boggling.
Yeah. Cool. I really like your system and the quality of the data.
Thanks. Yeah. It's cool to play with. I don't think we're finished yet, right? I think there's a few more things to learn from this culture system now.
So do we have any more questions for Andrew? Alrighty, I guess we will wrap up here for today. I'd like to thank our speaker Andrew again and everyone for your participation. So we will take a break next week and resume in 2 weeks. So take care, guys.
All right. Thanks so much, everyone.
Thank you. Bye-bye.
Nice meeting you.
Take care. Have a good day.
Bye.
Bye.